Hello, I'm Hannah Donner with the Collaborative on Health and the Environment. J&J is bringing you the latest environmental health science through our partnership webinars, Science Serves, publications, and social media. I would like to welcome everyone to today's CHE-ADC Strategies Partnership webinar, which is titled Obesogens and the Obesity Pandemic, a focus on prevention. Our moderator today is Janan Jensen, Executive Director of the Health and Environment Alliance, or HEAL. We will leave time following the speakers' presentations for a brief Q&A session. You may type in questions through the Q&A feature available in the menu bar at the top of your window at any point during the presentation. For those of you who have called in on the phone, we have posted today's slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on our webinar right now is muted with the exception of our moderator and our speakers. This webinar is scheduled to last for 70 minutes and is being recorded for our call and webinar archive. So with that, I'll turn things over to you, Janan.
Thank you very much, Hannah, and it's really great to be here on this webinar today. I'm very pleased to be able to present the three speakers we have today, and which actually they're going to be representing a compendium of 43 scientists from around the world, and a summary of this work from an international consortium. And the first speaker we will have is Robert Lustig, who is Emeritus Professor of Pediatrics in the Division of Endocrinology and member of the Institute for Health Policy Studies at UCSF. Dr. Lustig is a neuroendocrinologist, and he has expertise in metabolism, obesity, and nutrition. And he's also one of the leaders of the current anti-sugar movement that is changing the food industry. And secondly, we will have Jerry Heindel, who has a PhD in biochemistry from the University of Michigan. And he's worked in the area of reproductive biology and toxicology while on the faculty of the University of Texas Medical School at Houston and the University of Mississippi. Later, he was a scientific program administrator responsible for developing and administrating the NIEHS grants program in endocrine disruption, the development basis of diseases and obesity and diabetes. He is now retired from NIHS and is currently the director of the Commonwealth Program, Healthy Environment and Endocrine Disruptor Strategies. And the third speaker in presentation, we will be coming from Chris Kassotis, who is an assistant professor at the Institute of Environmental Health Sciences and Department of Pharmacology at Wayne State University in Detroit. Now in his independent laboratory, he is pursuing novel environmental sources of endocrine disruptors and/or metabolic disruptors, examining the mixture effects from combination of these chemicals and determining underlying molecular mechanisms. So with that introduction, I would like to give the floor for our first presentation, Obesity I, the overview and molecular and biochemical mechanisms. And for that, Dr. Lustig, the floor is yours, and a warm welcome.
Thank you so much, Janan, and welcome to everyone listening. So before we talk about obesogens, we actually have to talk about obesity. And that's my job, my task today. People think that obesity is because of a defect in energy balance. That is, you eat too much, you exercise too little, therefore you get fat, therefore you get sick. That is not exactly correct. And I'm going to give you some slides that basically bring us into the realm of biochemistry and the biochemical forces that are at work here. Now, this is a Venn diagram of the entire US adult population, 260 million. We are now up to 42.4% obesity, according to the CDC. That would mean 57.6% normal weight, BMI over 30, BMI under 30. Now, the standard mantra from the doctors and the nutritionists and the dietitians and the White House and the food industry and Congress is the following. 80% of those obese individuals, 80% of 42.4%, these 86 million people here, they're sick. They're fat and they're sick because they're fat. And if they would only just diet and exercise, we could solve this problem. This is not true. And the reason it's not true is because, in fact, 60% of the normal weight population have the exact same diseases as do the obese. Normal weight people get type 2 diabetes, hypertension, dyslipidemia, cardiovascular disease, cancer, dementia, fatty liver disease, polycystic ovarian disease also. When you do the math on that, on the two together, that's 176 million out of 260 million sick. And it turns out there are actually more thin sick people at 90 million than there are fat sick people at 86 million. So the whole concept needs to be rewritten, because if normal weight people get it too, how can it be about behavior? And I can prove that. Here we have two equally weighted individuals. Notice trunk fat 12.8, 12.8. One's healthy, one's sick. Which one's sick? The one on the bottom, B. And the reason is because the one on the top, he has subcutaneous, or if you will, politically incorrect, big butt fat, if you will. This guy down here has big belly fat or visceral fat. And we have a name for this. The visceral fat is much more metabolically active. We have a name for this. It's called TOFI, thin on the outside, fat on the inside, real medical term. Here's another example of the dissociation of obesity and metabolic disease. These are the little women of LOHA. These are a founder effect growth hormone receptor deficiency, in other words, Laron dwarfism, that are found in Ecuador. And when you take a look at their risk of cancer and diabetes compared to their relatives who do not have growth hormone receptor deficiency, even though you can clearly see they are all obese, their risk for diabetes is zero. Their risk for cancer is zero compared to their relatives. Okay, and you can see they're markedly obese, but they do not get any of these diseases. In addition, even though they're obese, their insulin levels and their HOMA-IR, that is their level of insulin resistance, are markedly decreased compared to their wild-type peers and relatives. And when you look at their biochemistry, you can see their percent fat is way up, so clearly obese. You can see their fasting insulin, however, is 1/3 of that of their relatives, as well as their HOMA. And you can see their LDLs are higher, but their triglyceride, their HDL is also higher, and their triglyceride is actually much lower. So here's their glucose tolerance test, and you can see their glucose levels are about the same, but take a look at their insulin response. And it is that insulin response that drives obesity normally, but not in these patients. So clearly, this disorder differentiates obesity from disease. Well, there's another disorder on the opposite spectrum, and that is that of lipodystrophy. These patients don't have any place to put fat. They have absence of limb fat. They have a normal or excessive facial fat because there's no place to put the fat. They have Cushingoid facies. They have a dorsocervical fat pad. They have acanthosis nigricans because their insulin levels are in the hundreds to thousands, and they have the most severe metabolic syndrome of all, despite the fact that they actually have less fat. The reason for this dichotomy is because not all fat's the same. Not all fat responds the same. Not all fat means the same in terms of disease. In fact, there are 3 fat depots. The first is subcutaneous fat. Now, you can eat and you can gain weight, and so your fat cells can grow and you can store more fat. In general, that actually is protective. That's where your body wants to put excess energy, is into those subcutaneous adipocytes for storage. Now, if you grow that fat so severely that the perilipin border of the fat vacuole inside those adipocytes decomposes or gets stretched to the point of no return, then the adipose tissue will basically become necrotic. You will get macrophages moving in. You will end up increasing the cytokine production, and then that will affect the liver. That will lead to increased hepatic gluconeogenesis. That will increase blood glucose, thereby stimulating islet cells, and so you will end up with further hyperinsulinemia. So you end up with this vicious cycle. And then the muscle is along for the ride. This is one model. This is the standard model of insulin resistance that doctors learn in medical school. However, as you can see in this slide, we're looking at body mass index, or an index of obesity on the x-axis, against insulin sensitivity here on the y-axis. And you'll notice there is a hyperbolic relationship, which has been known for years, first noted by Dr. Richard Bergman at USC. But what you'll notice is that for any given body mass index, there are patients who are insulin sensitive, and then there are other patients who are insulin resistant. So what that's telling you is that BMI is not a particularly good marker for obesity or disease. In fact, insulin resistance is a much better marker for both diabetes and cardiovascular disease, as you can see here. So it doesn't matter what the BMI is. The insulin resistant patients are much sicker than the insulin sensitive patients in both cases. And if you look at mortality, you'll notice it's not the obese versus non-obese that makes the difference. It's the insulin sensitive versus the insulin resistant that makes the difference. So disease and obesity, while they overlap, they clearly are very different. And so subcutaneous fat is actually not the big issue, but that's the one that you measure on the scale. Now, let's take visceral fat. Visceral fat is the fat in your belly. Now, normally, the visceral fat is the metabolically active fat. And the reason is because that visceral fat will, when it releases its lipid, it goes straight to the liver for through the portal vein rather than into the systemic circulation. So it is what we would call more metabolically active. Now, when you turn on the adrenergic nervous system, either from acute stress or exercise, you will then increase sympathetic output of norepinephrine, which will bind to the beta-3 adrenergic receptor on the adipocyte, which will lead to lipolysis. And also you might increase the brown fat to increase thermogenesis. And so basically, the sympathetic nervous system will lead to maintenance of body weight. In fact, you will burn fat and lose weight. However, when you are chronically stressed, you have an added factor of cortisol, and cortisol basically makes visceral fat take up more energy. And in addition, you also have the output of a cofactor from those adrenergic nerve terminals, neuropeptide Y. The neuropeptide Y binds to the Y2 receptor and actually inhibits that beta-3 adrenergic receptor from functioning. In other words, you can take a process that normally would cause lipolysis and turn it into a process that promotes lipogenesis instead, because of this adrenergic cofactor NPY. And then finally, we have ectopic fat or liver fat, fat where it doesn't belong, and particularly in liver and to some extent muscle. So here is normal liver here on the left side, and you can see the sinusoids and the Kupffer cells and the biocanaliculi all nicely ordered. And over here on the right, you have what we would call fatty liver disease, and you can see the fat vacuoles, and you can see the macrophages coming in, and you can see the beginning of scarring here and fibrosis. The question is, what's the substrate that led to this? In most cases, prior to 1980, this would have been alcohol, but now it's other substrates, and in particular, as you'll see, sugar. Now, non-alcoholic fatty liver disease is now the leading cause of liver transplant in the United States, having overtaken hepatitis C. This is truly now the silent killer, even having taken over from hypertension. And you would say, well, NAFLD is a problem of the obese. Actually, that's not true. It's a worldwide problem, even in normal weight people. So here you can see the NAFLD prevalence in overweight people. Yes, fair, it's much higher, but there is a NAFLD prevalence even in normal weight people and throughout the world. Okay, and if you look and if you bin the BMIs here on the x-axis, here's the hazard risk ratio for liver fat, and you can see that there is a signal even in the overweight and even in the normal weight population, clearly much less than the obese, no argument there. But the other thing that's really important is that with respect to diabetes, even in the normal weight population, if you have liver fat, you are 3.5 times more likely to develop diabetes in your lifetime, and normal weight. So very clearly, liver fat is the most egregious of the liver fat depots. And you can see here, adolescents never had liver fat before, And now 25% of all adolescents, not obese adolescents, all adolescents now have liver fat. And the hazard risk ratio for developing diabetes, if you have liver fat as an adolescent, is 2.6. So very clearly, liver fat is the bad guy. And as you will learn from Dr. Heindel in just a few minutes, a lot of the obesogens target the liver. So here's a way to think about this visually. Here we have an MRI fat fraction map of an obese person. Notice the love handles here on the side, but I want you to take a look at this guy's liver here. 2.6% liver fat. This person has MHO, metabolically healthy obese. Nothing wrong with this guy. This guy will outlive you. Now, take a look at this guy. Now, here you see obesity, of course, but now take a look at this liver, 24% liver fat. Okay, this guy's got non-alcoholic fatty liver disease. This guy's got metabolic syndrome. Now take a look at this guy over here. Notice normal levels of fat on the subcutaneous tissue, but take a look at this guy's liver, 23% liver fat. Thin sick, fat healthy. So again, the concept is it's not the fat you can see that contributes to disease, it's the fat you can't. So another way to think of metabolic disease and insulin resistance is that the problem, instead of starting in the adipose tissue, actually starts in the liver, with fatty liver leading to insulin resistance, thereby causing the beta cells to have to work harder to make the liver do its job. That raises insulin levels all over the body, driving further hyperinsulinemia, and that's what ends up storing the fat. And then, of course, the muscles along for the ride. But the liver is the primary fat depot. Now, what we have learned about the development of fat stores is that there are adipose tissue transcription factors, the most important of which is PPAR gamma. And this is the one that basically turns stem cells into adipose tissues, and particularly subcutaneous fat cells. This is where, for instance, thiazolidinediones work. And it's the reason why TZDs are associated with weight gain, is because they are actually recruiting pre-adipocytes to become adipocytes. But there are others as well, including other PPARs and retinoid X receptor, which dimerizes with PPAR to form to generate the liver fat to generate the subcutaneous fat signal. But there are also these liver transcription factors as well, and there are different obesogens that impact each of these different receptors to drive liver fat and thereby chronic metabolic disease. In addition, there are also endocrine receptors, such as the insulin receptor. There is no weight gain without the insulin receptor, as has been proven time and time again in type 1 diabetics. But there's the estrogen receptor, which increases subcutaneous fat. There's the androgen receptor, which inhibits fat development. There's the glucocorticoid receptor, which drives visceral fat development. And then finally, thyroid hormone receptors involved in thermogenesis. Now, one of the big questions is, why does the brain not take care of this problem? And the reason is there are all these signals coming from all the peripheral tissues, but they then get integrated at the level of the hypothalamus. Okay, and the hypothalamus has two separate pathways, one for orexigenesis, that is, eat more, and the other one is for anorexigenesis, that is, eat less. And the question is, obviously, there's something wrong here, and what is wrong? And the answer is these two hormones, leptin and insulin. And the problem is that leptin and insulin on those hypothalamic neurons crosstalk. And when they crosstalk, it turns out hyperinsulinemia inactivates leptin signal transduction, and particularly through SOX3, also through IRS2, and also through another one, PI3 kinase. So bottom line is, the higher the insulin goes, the less well your brain can see its leptin. And in that case, then your brain thinks you're a leptin deficient because you're not getting the leptin signal, which then causes you to eat more and burn less. Lastly, obesity starts in the womb. And we know that because of studies of small for gestational age patients who become obese and develop metabolic syndrome. And this is through a process called developmental programming, because the brain basically learns that for some reason, nutrition is hard to get, therefore store everything. It happens in large for gestational age patients due to epigenetic problems where basically certain genes are turned off or turned on. Prenatal stress and glucocorticoids, thus creating increased visceral fat depots right from the start. And finally, what we are going to talk about the rest of today, environmental exposures. And you will learn about all of these coming up now with Dr. Heindel's talk on the specifics of obesogens. So take it away, Jerry.
Okay, thank you so much, Dr. Lustig. That was a fantastic presentation, really broke things down biologically. While we're waiting for Dr. Heindel to pull up his slides, I'd remind everyone in our audience, you can put in questions in our Q&A feature on Zoom, and we'll get to them at the end of all three of our presentations. All right, Jerry, take it away.
Okay, thank you very much. And thanks, Rob, for that terrific introduction. And now we're ready to focus on obesogens. And I'd just like to start by showing all of the co-authors to the paper that we wrote. It had over 43 authors from around the world, over 20 iterations of this thing over 15 months. But finally, the paper is now out, and I thank all of them for all of their hard work. So the question is, what's causing obesity? And if we want to know that, way to look at it is what's changed in the last 50 to 70 years. That's the time frame when obesity really took off. And two things that aren't really big players are genetics, because genetics haven't changed in the last 50 years, and really exercise. It plays a small role because there hasn't been a lot of change in exercise. What's really changed is nutrition, meaning our diet has changed, and obesogens. They've come to play in the last 50 years or so, and everything kind of fits. The timeline fits. These chemicals were produced and put into play 10 or 20 years before the obesogen epidemic took place. And then in addition, there's an epidemic of obese 6-month-old babies, which can't really be due to diet. And obesity in all these rodents and domestic pets, which really isn't likely due just to changes in diet. There's too many different organisms here that are affected. So let's look at the current focus. The current focus of the clinical community is on overeating as the cause of the obesity epidemic. Therefore, their focus is on drugs, diet, and surgery. And if that was working, there should be a decline in the rates of obesity. But the global obesity rates are increasing, especially in infants and children. Therefore, something is missing. An important contributor to obesity, something that can focus on stimulating overeating. And we propose that obesogens are the key. They modify eating behaviors, and they can provide a really needed focus on prevention. Instead of waiting till people are obese and giving them diet, drugs, or surgery, let's focus on preventing the obesity in the first place. So this is the slide you've already seen. And the critical thing here is that this is endocrine control of all of these aspects of metabolism. So it's all hormones that are going back and forth between these tissues, integrating all of the tissues and the signals, controlling metabolism and weight gain. And if that's the case, then all these hormones and these hormonal pathways are potential targets for obesogens, chemicals that can cause weight gain. And they can affect all of these different areas. But let me go back and set the stage first. So there are a lot of chemicals in commerce. Most of them aren't toxic. Some of them are going to be toxic because they just kill you. But some are toxic because they interfere with the endocrine system, and they're called endocrine disruptors. So an endocrine disruptor is an exogenous chemical or mixture of chemicals that interferes with any aspect of hormone action. These are chemicals designed for a specific purpose, but they have a side effect that they mimic or antagonize hormone action. And there are about 1,000 of those that we know. But some of these endocrine disruptors have a very specific effect that they affect metabolism, and they stimulate weight gain. And they're a subset of endocrine disruptors, and they're called obesogens. Now, since obesogens act via the hormones and hormone receptors as either agonists or antagonists, they use the same principles as hormones. So they're going to be tissue specific, sexually dimorphic, act at very low doses, have non-monotonic dose responses, which means that in many times the effect at low doses will be different than the effect at high doses. And also the effects are life stage specific. You can have effects across your lifespan, but development is the most sensitive life stage. And you also have a lag time between the exposure and the weight gain. If the exposure is during development in utero, a lot of times you don't see the weight gain until you're 5 or 6 years old, or maybe even an adult. So here's the obesity paradigm. The obesity pandemic is due, at least in part, to exposure to endocrine disrupting chemicals during development. These chemicals, called obesogens, act during development to interfere with adipose tissue development. They increase the number of fat cells and the size of fat cells. They also interfere with the control of food intake and metabolism, because they can have effects on the development of the pancreas, liver, GI tract, brain, and muscle. And because of these effects then, that they're altering the programming of obesity set point, or they alter the sensitivity for developing obesity later in life. Now, we know that food intake and exercise are playing a role here, but obesogens can alter this set point for gaining weight. That is, how much food does it take to put on weight, and how much exercise is needed to reduce weight. And they do that by altering the programming of the metabolic system in utero when the tissues are developing. So the general scheme of obesogens, that these are chemicals that interfere with the normal hormone signaling pathways and alter the number and size of adipocytes, alter food intake. They can alter your microbiome, and actually there are many more places they can act. You can see here, if you look around the circle, there are all these things that are disrupted by obesogens, including causing fatty liver and skeletal muscle and glucose and leptin intolerance, insulin resistance. But I don't have time to talk about all of that today. I'm just going to talk about what's in the red: adipocytes, body weight, food intake, and microbiome. So where do we get these obesogens from? They're everywhere. They're in the air, they're in the dust, they're in the food, they're in the water. They can come in through your skin. We all carry a burden of many, many obesogens in our body. And you can see they come from disinfectants, pesticides, solvents. They come from your clothing, food preservatives, personal care products, everywhere you look. So the bottom line is at birth, we're already born pre-polluted with many of these obesogenic chemicals, setting us up for increased sensitivity for obesity throughout our lifespan. Now, clinicians already know about obesogens because there are antidepressant medications that will increase weight. There are blood pressure medications that increase weight. And there are diabetes medications, some that increase weight and some actually here that decrease weight. And the ones that increase weight, as you just heard, they activate the PPR gamma RXR pathway and cause increased number of fat cells, the same pathway that some of these obesogens like tributyltin activate. And then finally, there's dibutylstilbestrol, which was given to pregnant women to inhibit the pregnancy loss, didn't actually work. And in animals, look what happens when you give diethylstilbestrol early in life. And this happens at the very low dose of diethylstilbestrol and not at the high dose. So the second principle or proof of principle here is smoking. Smoking during pregnancy increases the weight of children at about the time they go to school. And you can see here from no smoking to more smoking to more smoking, they get more and more overweight. And on the left here just shows that all the studies are to the right of this line, indicating all of these studies, no matter where they come from and what kind of studies they are, they all show that smoking during pregnancy will increase weight of children. So we have very strong human data, and there's also strong animal data from smoking and also from the chemical that we believe is the main culprit here, nicotine. So this is a list of the obesogens with the strongest supporting data, meaning there's in vitro cell culture data, small animal data like zebrafish, rodent data, and human data. So it's nicotine, it's air pollution, DDT, bisphenols, bisphenol A, and in red here it shows that because we know bisphenol A is an obesogen, they came up with different chemicals to replace BPA, and guess what? They're all obesogens too. Phthalates and tributyltin. But there's a whole list of additional obesogens that have significant weight of data, but just not quite as strong as the ones I just showed you. Perfluorinated compounds, the forever chemicals, the PFAS, flame retardants. And again, we have these flame retardants, PBDEs. They were replaced with new flame retardants, OPFRs, and they're also just as toxic and obesogenic. We have fructose and non-nutritive sweeteners put on weight. A lot of pesticides will cause weight gain. Food preservatives, additives, and emulsifiers, monosodium glutamate, cadmium, arsenic, and dibutyltin. And here's something I think is really important. So when we talk about the diet is important, this Western diet itself is obesogenic. This high fat, high sugar, high salt, low in fiber, high in processed foods, inadequate fresh fruit and vegetables is obesogenic by itself. As bad as that is, if you're having this Western diet, you are also being exposed to a whole list of obesogens because of the diet and where the diet comes from and all this processed stuff that's in the food. You have BPA, phthalates, perfluorinated, fructose, all of these emulsifiers, MSG, and pesticides found on the fresh fruit and vegetables. So you get a double whammy when you're on this Western diet, which of course most of us are on. But it's even worse for a subset of the population, the people who are low income and minority populations. So they are having the Western diet, but also because of where these people live, they have high concentrations of PCBs, pesticides, phthalates, air pollutants, and BPA. Now, there's also potential obesogens. A potential obesogen is one that really has just been tested in cell culture, or it might have one small animal study. So there are assays in culture where you can start with a fat cell, and you'll hear more about this later. You start with a pre-adipocyte, add different chemicals, and if they form normal adipocytes, they're potential obesogens. And you can see here we have glyphosate, some pesticides, even house dust extracts. And DINCH in red is a replacement chemical for some phthalates, which is also still an obesogen. Now here's something that I think it could be really important. These chemicals cause an increase in fat cells. And when 3 of them were looked in more detail, all the genes that get turned on when they become fat cells, what happens is, compared to rosiglitazone here, which people believe makes a normal fat cell, the tributyl and also BPA and DEHP, a phthalate, make fat cells, and the genes that are turned on are not the same. They make what's an immature or incompletely differentiated fat cell that has all kinds of problems. So this could be part of the problem that these obesogens are causing obesity. Not only do they make more fat cells, but they make dysfunctional fat cells. Now, all the different obesogens haven't been tested. So which chemicals will produce these dysfunctional adipocytes? We don't know, but 3 of them here we know. Now let me go through a couple of the obesogens and show you some data. Prenatal exposure to air pollution, both the organic component, the PAHs, and the particulate matter, PM2.5. Each of these separately, when tested, are obesogens. And here's PAHs causing increased weight gain after exposure in utero, both in males and females. Down on the bottom left here, you see the increase in size and number of fat cells. And if you look in the human situation, the PAHs, the more you increase PAHs during pregnancy, the increased weight gain of the offspring at age 5 or 7. And the PM2.5 has the same effects. So you have in vitro data, strong animal data, and some good human data on air pollution. And we can take a quick look at some phthalates, which are plasticizers that are found all over the place in plastics and toys and packaging. In vitro in the cell cultures, you can see compared to the normal here, you see all the color due to the fat cells. And even in adults here, adult exposure to these mice increases food intake and increases the fat cells. You can see the fat cells here compared to normal. And if you look on the bottom, you see the increased number and size of fat cells. Now, one thing that's important to look, if you look across where the circle is here, you see there's more fat in this lower dose than the dose to the far right, the higher dose. So you're seeing here again, a non-monotonic dose response curve where the highest effect is at the low dose. And this works in developmental exposure also, where the low dose in the circle here is having the highest effect. And if you look in human studies, developmental exposure, and then they're looking at weight at 12 years, you can see several of the different phthalates as you increase the concentrations. At 12 years, the offspring have increased weight. So with regard to obesogens, you have strong in vitro and animal data and some pretty good human data. And then there's BPA. Everybody knows that BPA is an obesogen, and on the left you see female mice were given BPA during pregnancy and postpartum, first couple of weeks. And if you follow over the lifespan, you can see in red, the low dose of BPA increases weight compared to the control blue down here. You also see that it's non-monotonic. The lower doses are having a bigger effect than this high purple dose, which is not different from the control. Now, if you do this experiment in a rat, it's a little bit different. You do get increased food intake, and you can relate that, the increased weight, and you can relate that to increase in food intake. And if you go look in the brain, you can see that the BPA has actually increased the number of appetite neurons and decreased the number of satiety neurons in the brain, which is a nice mechanism for the increased food intake. And of course, it works in vitro. Look at all the fat the BPA puts in these cells. It makes these pre-adipocytes fat cells. So with regard to BPA, we have very strong in vitro and animal data. Unfortunately, the human data is a little inconsistent because BPA is such a short half-life. It's hard to get a good value for the human exposure over pregnancy time. And lastly, organotins, which are fungicides. You can see they put on increased fat here, and they do it at extremely low doses, nanomolar doses in the animals and in culture. And if you look at the bottom left-hand side, you see that these are HMCs, these are pre-adipocytes that you're adding tributyltin in the green, and you see it increases fat. But at the same time, these mesenchymal stem cells could be either fat cells or bone cells. And when you add tributyl-T, you make them become fat cells at the expense of bone cells. And of course, here you can see the color, the tributyltin increasing the fat. So here again, we have strong in vitro and animal data. Unfortunately, there's minimal human data. So I'm going to go over some general things here that came out when we wrote the paper. There are obesogens that specifically stimulate food intake that provides a mechanism and a pathway for the increased weight. They're BPA, DEHP, phthalate, tributyltin, and this newer flame retardant. And all of these affect this pathway that stimulates food intake and decreases the satiety part. Now, in addition, there's another part of the brain that controls eating behavior that's called the hedonic pathway. That controls emotional eating, food addiction, and craving. And it looks like BPA and BADG are associated with binge eating and reward or emotional eating. And I think the more we look, we're going to find more chemicals affecting not just the satiety aspect, but this craving, emotional eating, which you see a lot of times in overweight people. The other part of this is the interaction with diet. The obesogenic, high-fat Western diet already causes weight gain. And these chemicals cause weight gain. But when you put the high-fat diet with these chemicals, you get a larger effect. And this is just phthalates, a pesticide, and nicotine to show that that indeed is the case. But here's an experiment that I think is very interesting, and again goes to the human situation. This is tributyl-10. Now, turns out this experiment is in what's called the F4 animals. These animals have not seen tributyl-10 for 4 generations. Only their great-great-grandmother saw tributyl-10. So as they're growing up, if you look down here in the red circle on the left-hand side, as they're growing up, their weight is normal. But even after all these generations of not seeing tributyl-10, if you put them on a high-fat diet, just a little bit of high fat, their sensitivity to gaining weight appears, and they gain more weight than the controls. So they're extra sensitive to gain weight. Then you take the fat diet away, they have a hard trouble losing that weight. Then if you go up to the top right-hand side, what you see is if you take these animals and put them on fasting, they lose weight. But if they were treated with tributyl-10 4 generations before, they don't lose as much weight. So they're resistant to fat loss. Oh my goodness, that looks just like the human situation. There are also some obesogens that affect the microbiome, and we're learning more and more about the microbiome and how it plays an important role in controlling weight. So look at all of the chemicals that, as part of what they do, they also affect the microbiome. And here's some data that I think could be really important. We say people are obese because they overeat. Well, here's 2 chemicals, DES and chlorpyrifos, that cause obesity. You saw the DES in the animal model, but they did it without increasing food intake. They're affecting metabolism, but not increasing food intake. And I think that could be really important. And finally, something that's also really important is this transgenerational epigenetic inheritance, where you feed a chemical to a pregnant mother, the offspring gets exposed, and then you take those offspring and you mate them for the next generation and the next generation. So you get down to generation 3 or 4 that's never seen the chemical. But the chemical has had effect to cause obesity in the first generation. And guess what? It also causes obesity in the 4th generation that has never seen the chemical. Tributyl-10, DDT, BPA, BPA phthalate mixture, and even a hydrocarbon mixture. So I think that's something very important, that obesity can be transferred across generations due to chemicals. Now, we don't have human data on this except for one piece that's really important, just came out. They measured DDT in grandmothers. They measured this in these mothers that were in the 1950s, and now they're grandmothers. And the granddaughters at age 26 have increased obesity if their grandmothers had high DDT. And if you know anything about epidemiology studies, this odds ratio of 3.6 is absolutely huge. So it's a very strong effect over 2 generations in humans. So what we have here, I think, is the perfect storm for obesity. During development, exposure to these obesogens and poor nutrition that alters your programming of your brain satiety centers, your fat cells, your general energy metabolic rate. And then throughout life, you've got an abnormal system, maybe with abnormal fat cells that I showed you, and you add to that high fat, high sugar diets and an altered microbiome and continued exposure to obesogens, and you get the perfect storm leading to this epidemic of obesity and metabolic diseases. But there's good news here. The good news is we should be able to prevent obesity. If during development, we can reduce exposures to the obesogens and improve nutrition, we can stop all this bad developmental programming. And then if on top of that, throughout life, we can improve nutrition and continue to reduce exposure to obesogens, we should be able to have an important effect to prevent obesity and metabolic diseases. So where are we today? We know there are chemicals that are obesogens. We can actually show a causal link between in vitro exposure and in vitro and animal exposures and weight gain or increased numbers of fat cells. And we can show collaborating associations in human studies. And really, that's the strongest data we're ever going to be able to get. We know we're highly exposed to these chemicals. We can tell in many cases when they act. We can show the mechanisms and pathways that they act. We can show they interact with diet to increase the effects of both of them. And we know the effects of diet, surgery, and drugs aren't working. So something else is needed. And perhaps we need to use this precautionary principle and add a focus on prevention, focused by improving nutrition and reducing obesogen and exposures during pregnancy, early childhood, and across the lifespan. So thank you very much.
Great. Thank you so much, Dr. Heindel. While we're waiting for our next speaker, Dr. Kassotis to join us, I would just remind everyone, you're welcome to put in questions in the Q&A. It looks like we have just one right now, so feel free to start adding your questions as we move forward, and we'll get to them after this final presentation. Thanks, Chris.
Thanks, Anna. So thanks everyone for being here. My job is to talk to you a little bit about the assays that we have available, both currently and in development, to evaluate and kind of prioritize obesogens. So first, I want to just highlight again that this is another great collaborative effort, right? So this third review paper brought together 21 authors from across the US, France, Sweden, and Australia. And so this was, brought together a lot of different expertise, really a strong collaborative effort. So this third section of our review highlighted the primary models of evaluating obesogens, both in vitro and in vivo. While there's been a reliance on some classical models of obesogens, I think there's a push for developing new in vitro and in vivo models, some of which are highlighted here. I'd like to think that we're sort of in a little bit of a renaissance right now for obesogenic chemical evaluation, with a lot of shifting standards, lots of new models, and lots of new data contributing to really both our functional understanding of obesity as well as our technical understanding of how chemicals may exacerbate or promote obesity. And so I'll spend the next 10 to 15 minutes overviewing some of the current state of the Obesogen model field, introducing some of these models, but really just very briefly in the interest of time. So just a note up front, so evaluating causal toxicity of chemicals really relies on a combination of both in vitro and in vivo models. And given, I think something that was highlighted well by our previous speakers, we really need good, robust, high throughput methods to accurately screen for and prioritize higher order testing, because we can't possibly test the tens of thousands of chemicals in commerce in rodent models for metabolic disruption or obesogenic effects. And then we need more reliable in vivo models that are cost effective, have high translation to human health, and importantly are well validated. And traditionally, metabolic disrupting chemical research or obesogenic chemical research has relied heavily on rodent-based cell and animal models, right? So the 323L1 mirroring pre-adiposite model and also rodent testing, of course. But that's been broadening over time. So we have increasing use of mesenchymal stem cells and also increasing use of human cell models, both MSCs as well as pre-adiposites. We have increasing use of fish models. We spent some time in this review talking about both the zebrafish and Medaka. And then also some non-traditional obesogenic or models such as the fruit fly or C. elegans. So I won't belabor this. There's certainly a spectrum of avenues for chemicals to disrupt metabolic health, and that's been covered well by our previous speakers. But just important that the next few slides I'll focus on more of these in vitro effects of commitment and subsequent differentiation of adipocytes. So just briefly, if we start from this multipotent mesenchymal stem cell source that can differentiate down several pathways, we can certainly push cells down the adipocyte pathway, but also myoblast, osteoblast, chondroblast, etc. Many of the in vitro models target the committed pre-adipocyte cell stage early in the differentiation process. So this is where our 323L1 model comes in, but also several of the other kind of commonly applied models here. And in this case, activation of several early stage adipogenic genes, PPAR gamma, the CCAT enhancer binding protein family, are the things that are going to spur subsequent differentiation and push them out of this kind of immature precursor fat cell stage. And as they develop, you're going to start accumulating lipids around the periphery of the cell, and eventually, have kind of a packed cell crammed with lipids, at the end of your assay. So really kind of the gold standard in vitro testing has been the 323L1 pre-adipocyte assay. So these are Swiss albino mouse embryonic fibroblasts, committed pre-adipocytes. So they are not going to develop down other cell lineages. And they've really been used extensively for decades to evaluate adipogenesis. We understand the mechanisms that drive adipocyte differentiation very well in this model, and it's proven to be quite reliable for screening metabolic disruption in vivo. And then there's just a little bit of a timeline here for you, but it's about a 2-week assay where you're seeding plates, letting them grow to confluency, giving them 48 hours to undergo growth arrest and revert to clonal expansion, at which time you can initiate differentiation, generally with a differentiation cocktail of some sort. Generally that's achieved with IBMX and fetal bovine serum and some insulin and sometimes some other factors. Often that's when test chemical exposures start as well. That's a 48-hour window at which time you switch from differentiation medium to an adipocyte maintenance medium, generally removing the IBMX, retaining the fetal bovine serum insulin, and your test chemical exposures for another 8 days, and then at the end, assaying for your adipogenesis markers. And what are those? Those are, of course, triglyceride accumulation, generally achieved with adipo-red or Nile red dye, and then cell proliferation or cytotoxicity, which is generally achieved through some sort of DNA dye. And this is kind of just a little picture to put this in context, some 3T301 cells in the wild, so to say. So you've got some blue nuclear DNA staining here. And then the kind of yellow gold that you see is the intracellular triglycerides from some rosiglitazone or PPAR gamma agonist treated cells. So a good positive control in these assays. So our review focused not just on the adipocyte models, but also a growing use and reliance on liver and muscle cell assays to add to our understanding of obesogenic chemical evaluations and kind of broader metabolic disruption. And the use of varying hepatocyte models for evaluating things like non-alcoholic fatty liver disease and other metabolic disorders has been covered recently, certainly by others. These models have been used to evaluate kind of really diverse environmental contaminants, bisphenols, phthalates, pesticides, and other environmental chemicals. And then there's an increasing body of work looking at muscle cell assays as well, which I'll talk about a little bit more. So some of the ways that this is shifting, as I mentioned, there's an increase in commercial availability of human mesenchymal stem cells. And a growing reliance on those, I think, in the literature that we've been seeing, particularly in the last year or two. There's also a growing commercial availability of human preadipocytes. There's less reliance on donors, so less labs now have to seek out their own source of human adipose to isolate the cells themselves. They can now order them from commercial companies, and they can source specifically from males or females, lean or obese donors, diabetic or non-diabetic donors, subcutaneous or visceral depots, all sorts of different characteristics. So they can really ask more targeted questions now than really we've been able to historically without pretty extensive collaborative efforts to obtain fresh cells and isolate them in the lab. There's also an ability to examine the interplay of commitment across cell lineages, such as bone and adipose, muscle, et cetera. As I mentioned, increasing utility of liver cell assays to examine this TAFLD or NAFLD phenotype, and increasing use of primary human hepatocytes. Of course, all of these human cell lines have their limitations, but have seen increasing use in drug metabolism and other studies. And then some limited but increasing evaluation of myogenic differentiation and ability of MDCs to suppress signaling or development in that sort of context as well. So most models using human mesenchymal stem cells still use a cocktail that commits the cells to the adipocyte lineage and pushes the cells to differentiate. So then measures triglyceride accumulation or DNA content as they have historically with the pre-adipocyte models. There's a growing push for more human cell use though, and a growing appreciation for the greater inherent variability in human cell lines. So screening efforts with one may not be sufficient, for example, whereas this has historically been accepted really without appreciable reproach using 3T301 cells. So some recent research, a shout out to the Blumberg lab, really described some protocols for distinguishing assays to evaluate adipogenic lineage commitment and subsequent adipocyte differentiation. And so cells can be pre-treated with test chemicals prior to the differentiation cocktail exposure. And those pre-treated cells can be subsequently exposed to the differentiation cocktail and evaluated at the end of the differentiation window. And then the extent of triglyceride accumulation can be compared with standard adipogenesis plates. and chemicals with effects on commitment should have equivalent effects to those differentiated for the full 2 weeks, whereas cells without effects on commitment should not accumulate more triglycerides than say, a vehicle control. So this is really kind of expanding our ability to ask broader questions in the obesogenic chemical field. I know we have limited time, so we've also spent some time talking in this review about in vivo models. Really won't spend much time on rodents. Really, that's pretty well established. But we do spend some time talking about some factors that are really important, things like animal feed, which has, quite a lot of importance when it comes to obesogenic chemical testing, and several other factors that play in. And so I'll go through some of the other models just briefly. So zebrafish, really kind of a burgeoning metabolic model, probably of these ones that we've talked about in this paper, probably the best established alternative to rodents. So they have measurable adipose and adipocytes as early as 9 to 12 days of development, really originally in the pancreatic and abdominal visceral depots. 34 anatomically, physiologically, molecularly distinct adipose depots throughout the body of the fish. And the work by John Rawls' group at Duke has really established a clear developmental timeline for each of these depots, which is really helpful. Fish adipose is really quite similar to human. It separates the same way, but importantly, they don't have brown adipose. So one limitation of this model. So as I said, the molecular mechanisms underlying the adipocyte and lipid depot development are highly conserved. So a lot of the genes involved with adipocyte differentiation, endocrine function, lipolysis, et cetera, energy storage, all of these are highly conserved with across both the zebrafish and all the way to humans. The adipose depots respond to high fat challenge and food withdrawal as you would anticipate in the same way that we can see in rodent models. And while rodent models, imaging of whole body adipose is limited and generally low resolution and challenging, a high resolution imaging of all of the adipose depots in the fish is very easy. So a real strength of the model. C. elegans, won't spend a lot of time here, but really importantly, the main regulatory pathways of energy homeostasis are shared with mammals. You do generally see lower conservation of some of the pathways and some specific organs that are lacking in this model. They do lack PPAR gamma, importantly, generally thought of as the master regulator of adipogenesis, but they do have orthologs to PPAR alpha and PPAR delta. So definitely some important pathways that are conserved. No homolog for leptin, no cells specifically designed for lipid storage, so they don't have adipocytes, but they do store lipids primarily in intestinal epidermal skin-like cells. And there's been some work on things like some of the bisphenols, methylmercury, and some other metabolic disruptors to evaluate potential obesogens in this model. Drosophila, again, lots of advantages to this model. There is some functional overlap with humans despite some pretty large anatomical differences. The fat body covers many of the metabolic health functions of both liver and adipose tissue in a human. And we've seen some important testing for DHP, methylmercury, and some PFAS that have increased weight and adiposity or disrupted metabolic signaling in this model. And then lastly, just to bring this home, I just want to highlight that really there is a need for new and improved standardized testing methods to identify chemicals that disrupt metabolic health, and really through diverse mechanisms. And there's a lot of large-scale European Union efforts designed to address this gap. And so I'm excited to see where those go in the coming years. We really still need an improved understanding and validation of alternative and emerging in vitro and in vivo models. We've definitely seen an increasing use of these new emerging animal models, emerging for obesogenic chemical testing anyway, certainly well-established lab models. We see a growing use of use and availability of human in vitro models and some of these 3D and spheroid cell culture techniques. And lastly, that predictive modeling may offer some improved utility in screening kind of the myriad chemicals in commerce for obesogenic properties. We need reliable, reproducible toxcast and other input data. I do a little shout out. We just published a paper in Environmental Health Perspectives, doing some predictive modeling for obesogenic chemical evaluation. But really, this sort of effort needs a robust understanding of the molecular initiating events and the contributory mechanisms that drive the eventual development of obesity. And so I'll stop there. Thank all the co-authors again, everyone's attention, and I think we can now take questions.
Thank you. Oh, sorry, go. Hannah, did you want to come in? Sorry.
I wanted to say thank you so much and pass it off to you, Sian.
Okay, great. I mean, I am just, what a terrific presentations, and your ability to get that all into an hour. I think we have a lot of opportunities for prevention here in the European Union with the chemical strategy, because many of the obesogens that you mentioned, whether it's the phthalates or the PFAS or the bisphenols, are on the table for reduction and restriction. So that's really great to have this evidence to be able to bring to policymakers. So I would like to go to one of the first questions. First question I have is, does the route of exposure, such as oral versus inhalation or dermal, as in the case of occupational setting for obesogens, such as phthalates, does it have any effect on obesity? And I don't know who would like to volunteer to answer that. Jerry or Robert, perhaps?
Well, I can say something, I guess.
Mm-hmm.
It's likely the route of exposure would have an effect on the final concentration of the chemical in the blood and the tissues. But overall, probably exposure from any route would be effective. I don't think there'll be going to be much difference there.
I think it probably has to do with what the target is. For instance, if it's the liver, probably the route would probably be oral. If it's going to be, adipose tissue, it probably doesn't matter what the route is. It could be dermal, it could be pulmonary, it could be oral too. In fact, actually, oral might actually be preventative to some extent, because the microbiome might decide to chew it up instead. Ultimately, what we've learned about obesogens is there's not one obesogen, there's many. There's not one pathway to fat cell development, there are many. There's not one transcription factor, there are many. And that's great from a survival standpoint because you have redundancy in the system, but it's really lousy from a toxicity standpoint because lots of things are toxic.
Mm-hmm. Thank you. Another question, are the effects leading to obesity related to in utero exposures to obesogens exacerbated by exposures to the same ones later in life?
Okay, well, I don't know that we have data for that. Probably, I would expect so, but I don't know if we have data for that. Here's what I can say. We have data that shows in utero exposure to various obesogens correlates or predicts, I shouldn't say correlates, predicts obesity at age 3 years. And we also have data on those same obesogens in different patients promoting weight gain over time, over childhood. Childhood being a particularly vulnerable time for this, because that's the time of fat cell development anyway. And to be honest, and puberty is a very specific time for fat cell growth. So children are, the canaries in the coal mine when it comes to this problem. And it's likely that this is a primary driver of the childhood obesity pandemic that we see everywhere. So we don't have, shall we say, longitudinal data in individual patients, raw data to follow over time. But we do have data that says that there is an effects of these obesogens in terms of development, and we also have effect of those obesogens in terms of maintenance. So, yeah.
Thank you. Okay, I have another shorter another question on the, I think, Jerry, you talked about the transgenerational epigenetic effects through females, but not males, is the question. Are offspring of great-grandfathers not affected by great-grandfathers' exposure to obesogens? So I don't know if, Jerry, you want to answer that?
We don't really know
Yeah.
the answer to that. The experiments have all been done focusing on the pregnant mother with the exposure to the offspring.
Thank you. Any other comment? No? Okay. And
The answer is I don't know.
yeah.
What I do know is that you get your mitochondria from your mother. So if you have dysfunctional mitochondria from your mother, that's going to make a difference. Now, it's possible that genetics play a role in this, and that could be transmitted by the father.
Thank you. Yeah. Another question. Do you have animal studies where exposure was only in adulthood?
Yes, there are quite a few studies like that, and they show obesogenic effects. But what I'd like to see more of is a comparison of adult exposure to developmental in utero exposure, as far as the sensitivity and what are the actual effects and mechanisms. We don't have that very much, but there are certain areas like flame retardants where most of the experiments have been done actually in adults.
Okay. Thank you, Jerry. Here's maybe a question for Robert. What is the percentage of fatty liver disease in obese people compared to those that have normal weight?
Oh, well, I think I showed a slide.
Yeah, maybe you did.
I did. Without question, it is more common in those who are obese. And, that makes sense, because likely people who are obese have multiple liver fat, multiple fat depots that are increased. So, these things are not working in vacuums. And people who are obese, very likely are eating lousy, are stressed, and living in, shall we say, poor conditions that increase their risk for obesity. For instance, living close to freeways and with increased air pollution, all of those things, so the social determinants of health say, all three fat depots are going to be affected. And in fact, all, adipose tissue and liver differentiation is going to be affected all at the same time.
So I have I think a final question here. I mean, I know we're running a little bit over. I hope that's okay, Hannah, that we just have another question or two.
Absolutely.
Great. And I think it's stepping back to what kind of policies can help address reducing obesogens. I mean, I talked about a few in the European Union. Maybe you could talk about the public policy agenda in the US or globally. I don't know if anybody wants to take that one on, or maybe hear from Chris. I don't know if we've heard from you, if you wanted to maybe talk about testing. Yeah.
So we recently published some work just evaluating the regulatory environment globally around endocrine disruptors, so not even specifically obesogens. And it's very poor, right? So very few countries are doing a good job with regulating this sort of chemical. I'd say probably really no one is specifically looking at regulating obesogens at this point. So I think it's a real gap in the regulatory context, and I think it's something that we need to do better about. I think something that Jerry talks about often is the fact that really we're so focused on treating obesity, and we really need to start thinking about how do we prevent obesity in the first place. And one of the ways that we might do that is starting to regulate and limit some of these chemical exposures.
Right. I mean, the EPA has been really asleep at the switch on this entire issue. And, no individual can stop their exposure. Number one, they can't limit it because, it's in everything. And number two, they don't know that they're being exposed because all of this is invisible. So it really behooves a regulatory agency like the EPA, which was set up very specifically to do exactly this. I mean, it was supposed to be for air and water pollution, but hey, chemical pollution is pollution, and they should be taking the lead on this. And of course, they are not.
Thank you. I know in the European Union, they are considering the reauthorization of glyphosate. And I see that glyphosate was one of the obesogens that was mentioned in terms of having not as strong of evidence, but definitely having evidence. So that's another opportunity I see in the European Union.
Indeed.
Yeah. Okay, maybe Jerry, did you want to come in on that one as well, just in case you had anything to add for yeah, good. Okay, well, I see I've gotten through there's a few questions left. Maybe I can do one more. Here is one. Is there any difference in utero-effective obesogens in terms of gender of the developing fetus?
I don't believe so. I was the endocrinologist on the Chamacos study, which is the children and mothers of the Salinas Valley. We looked at, pesticides in the urine of pregnant itinerant worker mothers and looked at their offspring in terms of cognition, reproductive capacity, and also obesity. And to my knowledge, the obesity part of it was equal. There's no question from a reproductive capacity, there were clearly sex differences. The females showing earlier puberty, the males showing later puberty, as you might expect, almost assuredly because of the estrogens in the environment. But from a cognition and also from an obesity standpoint, I don't believe there was much of a sex difference.
Thank you very much. It's been a really amazing presentation, and the slide sets are available. And I also see that there was an article in The Guardian today about this review. So for those of you who want to have a summary, have a look there. And thank you all for being with us today. And I'll hand it back over to Hannah to close us up.
Great. Thank you so much, Shannon. I would just reiterate, I think the social determinants of health, meaning, your surroundings, your microbiome, everything that impacts you, everyone's exposure, there are disproportionate impacts on marginalized minority communities. And that really does need to be addressed because they really hit the hardest. So moving on from that, policies have to help those most impacted. All right. Well, thank you so much, Shannon, and to all of our speakers. We're approaching the end of today's webinar. A video recording will be available on CHE's website soon, and tomorrow you'll receive an email containing a link to the video. The next CHE EDC Strategies Partnership will take place on June 15th and will feature a new publication on exposures to PFAS and markers of liver injury. To learn more and to RSVP, please visit our website at healthandenvironment.org. If you are new to CHE and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate these CHE partnership webinars bringing the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers, Dr. Lustig, Dr. Heindel, and Dr. Kassotis for taking time to present today, and to you, Janan, for your excellent moderation. Thank you so much for joining us. We're wishing all much health and wellness. Thank you for all of your time and for all the work you're doing.
Thanks.
Bye. Thanks.
Thanks.
Thank you.